I'm Ewan Pearson. I'm Chair of Diabetic Medicine up in Dundee, and a member of the steering committee for the Pharmacogenetics and Stratified Medicine Network, and so chairing the afternoon in Munir's place. And I have pleasure in introducing our next speaker, who's thanks very much to the US Pharmacogenomics Research Network, who recommended him to us as a fantastic speaker, is Charles Paulding from Regeneron. And you can see the slides already up. So he's going to be talking us around polygenic risk scores and PCSK9 inhibition.
Okay, great. Thank you. Thanks for the intro. So yeah, I'm over at the US. So good morning to anybody else who's in the US, and good afternoon to everybody over in the UK. So today I'm going to talk about, I'll probably wind up touching on a number of things that other people had mentioned as I go through my slides. But the focus here is going to be a little bit more on drug response. And so I'll talk about using genome-wide polygenic risk scores, looking at our PCSK9 inhibitor, Alirocumab, in a large outcome study. Oops, let's see if I can get my slides to move. Okay. A little bit of background on the drug. So Alirocumab, this is a fully human monoclonal antibody that inhibits PCSK9, and this was co-developed by both Regeneron and Sanofi. Alirocumab is approved for the reduction in risk of MI, stroke, and unstable angina, requiring hospitalization in patients with established cardiovascular disease. Treatment with Alirocumab produces a substantial reduction in LDL cholesterol and other atherogenic lipoproteins. So in many respects, I think PCSK9 is kind of the poster child for using genetics in drug development. It had been discovered a number of years ago. Patients with gain of function mutations of PCSK9 have very high elevations in LDL cholesterol and also have very early onset of cardiovascular disease. And the reverse of that, patients with loss of function variants have a protective effect. They have much lower LDL cholesterol, as well as a much reduced risk for developing cardiovascular disease. So it's really kind of an ideal genetics supporting drug targets scenario for this particular drug. A little bit of background on the study and the objectives. So there had been some previous studies suggesting that patients with high coronary artery disease, or CAD, polygenic risk scores, may receive greater benefit from statin treatment. So we really wanted to see if can these polygenic risk score findings be extended to patients on PCSK9 therapy. And so the objective here was to test if patients with a high CAD polygenic risk score showed greater benefit with Alirocumab treatment. This is a touch on some information you've already gone over. I'll just go through this relatively quickly then. This is just the basic schematic for how we've been developing polygenic risk scores, very similar to what has been discussed in previous talks. Starting on the left with a very large, well-powered GWAS study. So we've been doing this quite frequently in our programs at Regeneron. And so we've done a number of these now, and we've actually developed a pipeline for developing polygenic risk scores. And it's shown a little bit in the bottom panel. I won't go into too much detail. We have a pipeline that, takes GWAS summary statistics. We can test it across a number of different polygenic risk score models, including pruning and thresholding, LD-PRED shown here. Now we've also incorporated some other methodologies as well. So we can basically go through and pick the best model going forward. And obviously you get a distribution through the population shown in the top panel there. And then finally over on the right, you get some population risk, on the x-axis, you have percentile of genetic risk, and this is looking at CAD and UKB. You can see patients with high polygenic risk scores have a much increased risk of developing cardiovascular disease. I think from our experience, really the best and most important component of all of this is really your starting source data set. You have very large GWAS data sets. To be frank, it's a little bit like shooting fish in a barrel now. Making these polygenic risk scores pretty much works every time. I will say, with regard to the differences in model, this was discussed by one of the speakers earlier, we don't see the same model sort of winning every time. Sometimes it will be LD-PRED, and sometimes one of the other models. And I think this probably gets to the power of your original GWAS, as well as sort of the genetic architecture of the particular phenotype you're looking at as well. So these are the data sets we use in generating our CAD polygenic risk score. So our source data set was the Cardiogram Plus consortium. This was 61,000 cases of CAD and 124,000 controls. And then we tested this using UKBiobank data set as our testing data set, and this is about 446,000 patients. Most of you are familiar with this with the link to the EHR. Then we also have access to our Geisinger healthcare system data set that we've been collaborating with the Geisinger health system to generate data there. And at the time of this study, we had 84,000 patients linked to the EHR. Now that's actually even larger. I think it's around 150,000. And so then finally, we test our model and we pick our model, and then we move to the clinical trial sample set, where I'll give more information on that. So all in all, in generating and testing this model, we have data from over 700,000 individuals, which is really pretty remarkable where genetics is going now with the access to these really large data sets. So one of the things we sort of tried to do too when we got to the clinical data set, as I mentioned before, we test a number of models, but by the time we get to the clinical data set, we really want to just have picked one model so that we're not potentially overfitting by choosing too many different models to test in our clinical trial sample sets. This is just the outcome of them and really just some of the data for what our polygenic risk score. Shown on the left is data from UKBiobank, and shown on the right is data from Geisinger health system. And this is looking at about 6 million variants on the right and 6.5 million variants on the left. And you can see on the x-axis, again, percentile of the genetic risk score. Patients in green to the left have low polygenic risk, and patients to the right have high genetic risk. You can see this has been shown in some of the previous talks, really high differentiation between people with very low risk and patients with very high risk with regard to cardiovascular events. And again, we see pretty similar results when we look at the Geisinger data on the right as well. Now we'll go into the actual analysis we did, looking at the Odyssey outcome study. And this was published about a year ago in Circulation. So if anybody wants more details, as I said, this is a fairly abbreviated talk here, so I could go into all the analysis that we did. So just one slide overview of the trial. So this was a large outcome study. Almost 19,000 patients were included in the study. They were included, inclusion criteria included patients having had a recent acute coronary syndrome, an MI or unstable angina, elevated LDL cholesterol, or elevated apolipoprotein B. The primary endpoint was MACE, which is really just a composite of cardiovascular events. And you can see on the right-hand side, this is a Kaplan-Meier curve showing the results of the trial. Overall, and the trial ran for a median of 2.8 years for patients in the trial. You can see on the y-axis is the MACE events or cardiovascular events, and clearly treatment with Alirocumab leads to a decrease in cardiovascular events over the course of the study. This was a successful trial in that regard with a hazard ratio of 0.85. Now, the pharmacogenomics study was conducted using, as I mentioned, the genome-wide polygenic risk score that I outlined a little bit in the previous slides, of about 6.5 million variants. And we looked at this in just under 12,000 patients for which we had DNA samples available. And really, for clinical trials, this is a very large, well-powered study to look at this particular thing. So the first question we really want to address is, looking at baseline, do we see differences in patients with high polygenic risk scores versus patients with low polygenic risk scores? And so I'll start off by saying, one has to define what is a high genetic risk score. And in this case, we took patients at the highest 10%, the top decile, and defined them as high genetic risk, and lower genetic risk was anybody below that particular threshold cutoff. So when you look through here, the differences between these two groups, high genetic versus low genetic risk. So in general, patients with high genetic risk tended to be younger. They tended to be female. Also, they had elevations in many cholesterol, LDL cholesterol, as well as apolipoprotein B. And interestingly, we found a really large difference in their lipoprotein A levels. Nearly double, more than double the median amount of this, and this was highly statistically significant, a p-value of 10 to the minus 56. And then down at the bottom of this, you can see this is the medical history, again, at baseline, showing that, again, across a range of these, family history and so forth, other cardiovascular endpoints here or conditions, there was an elevation in patients with high genetic risk. So this is pretty much what one would expect going into the study. So even at baseline, we can begin to see differences in these patients based on their polygenic risk. So the next question we wanted to address was, in the placebo arm over the course of the study, do we see differences in the number of events in patients with high genetic risk versus patients with low genetic risk? And the answer again is yes. You can see it here. So patients in red are shown with a high polygenic risk score, the top decile, compared to patients with lower genetic risk. And on the left-hand part of the panel here, this is the MACE. This is the composite of cardiovascular endpoints. And then on the right-hand side, we're seeing similar data for any cardiovascular disease. This is really just MACE, including the revascularization endpoint as well. You can see a hazard ratio of 1.59, and this was statistically significant for MACE. Again, showing then that over the course of the study, patients with high polygenic risk scores looked at longitudinally have an increase in cardiovascular events in the placebo arm. We next want to address really the main question of the study, and do patients with high polygenic risk scores receive greater clinical benefit with PCSK9 inhibition using Alirocumab? And the answer, again, for that is yes as well. Again, looking at some Kaplan-Meier curves. On the right-hand side, we see patients with high genetic risk in placebo and Alirocumab. And over the course of the study, you can see there's a pretty large decrease in cardiovascular events in patients treated with Alirocumab with a hazard ratio of 0.63. And this was statistically significant. Also was significant as an interaction term as well. And this is compared to patients with lower genetic risk shown in panel A with a hazard ratio of 0.87. Clearly, they're still receiving benefit. This was significant, but clearly it's diminished compared to patients with high polygenic risk scores. We then wanted to see what happens when we combine risk scores or risk of the study. It had been shown previously for this trial that patients who come into the study with very high LDL cholesterol, greater than 100 mg per deciliter, also had been shown to have greater benefit from taking Alirocumab. So we asked the question, what if we combined high genetic risk with high LDL cholesterol risk at baseline? Would we see an increase in benefit in those patients as well? And again, similar to what I showed in the previous slide, the answer is yes. So if you focus on panel B here, these are patients with high genetic risk and high LDL cholesterol. And so they have a hazard ratio of 0.55, even greater benefit than seen by either of these alone. Patients treated with Alirocumab have clearly a large decrease in cardiovascular events over the course of the study. And then panels A and D are sort of the intermediate phenotypes. A is high genetic risk and low LDL cholesterol baseline, and panel D is lower genetic risk and high LDL cholesterol at baseline. You can see these patients still get clinical benefit, hazard ratio 0.65 and 0.78. However, it's not as much as one sees when you have the combined risks of genetic risk with LDL cholesterol risk. And then finally, over in panel C, these are patients with low genetic risk and low LDL cholesterol. And you can see, they clearly don't derive as much benefit compared to the other groups here with these other risk factors. Very briefly, I know this has been discussed in some of the previous talks about the use of polygenic risk scores in varied ancestry groups. We did this study both ways, looking at just Europeans as well as including all patients. And it turns out it really looked pretty similar. And I don't have time to go through all of this, but basically you can check this out in the paper. We saw, although the sample sizes are small, we couldn't really do much statistics when we break out by different separate ancestry groups. There was a reduction in MACE with Alirocumab treatment, and really was very directionally consistent with high genetic risk patients across all the ancestry groups. So regardless of what their genetic background was, we see clinical benefit similar to what one sees in just a European-only analysis. So unbeknownst to us at the time, but when we published this in Circulation at the very same time, Amgen, some investigators working on the Amgen large outcome study, Fourier, also published a very similar study. We used different approaches. We used the genome-wide polygenic risk score. They used the 27 SNP score. And while there were obviously would be differences on how we conducted the studies, that were done completely independent, and we didn't know what they were doing, and they didn't know what we were doing. Nonetheless, these really replicated each other's findings in both of these studies, and these really are the two large outcome studies for PCSK9 inhibitors that have been conducted thus far. So patients with high polygenic risk scores received greater benefit really from PCSK9 treatment, whether it was either the Amgen drug or the Regeneron Sanofi drug, Alirocumab. So it was really nice to see this replication of our findings, and we essentially replicated each other's findings unknown to each other at the time. So a little bit on general considerations on polygenic risk scores. And some of this has come up in other talks. I've been asked, about, really using this clinically, putting this into practice, so to speak. And, the barriers are quite high for doing this for drug programs, because we have to really deal with the regulatory agencies in this regard. And I think there would be, it has the potential for it, but it's probably too early in that stage to do this. If you think about it, this would be a diagnostic test with 6.5 million variants. Most of these are imputed. I don't think there's a diagnostic test out there now that uses that type of setting. There'd be a lot of probably barriers. Also, one typically needs to go out and run a prospective study. These outcome trials in cardiovascular disease are hugely expensive and take a long time to run. So nonetheless, we decided to really keep this more as a research finding. So I do think going forward, it's a really exciting area. These scores are constantly improving. Every time a new GWAS will come out with more cardiovascular cases, we can continue to update the scores. And I think new methodologies are also being developed. As I said, since we published this, we're already incorporating a couple of new methodologies in-house when we test these as well. One of the things I didn't touch on that we're using internally quite a lot is gene-based or pathway-based scores. These can be a little more focused from the biology, and we're using this quite extensively as well. These can be quite valuable tools, research tools as well. I think for drug response, it's a little unclear as to how applicable these will be. There hasn't been a lot of cases like this where people are using this in pharmacogenomic setting. I know we're testing a lot internally for our other programs. I can't really speak to how that's going just now. But I do think, the cardiovascular disease case here might be a little bit unique. I think it really comes down to, again, your source GWAS data set. And the endpoint for this clinical trial is really the same one as the disease, right? It's cardiovascular events. And it was a very large, well-powered source data set. So I think that really helps power the study. In addition, this is a very large clinical trial, so it's well-powered in that response too. PR scores are, I think, this was touched upon as well. They're going to be quite widely available. They're really quite easy to generate based on common variation. One doesn't need to do sequencing. And from one array, you can generate any number of polygenic risk scores. And we're beginning to see this roll out with 23andMe and some academic centers as well offering polygenic risk scores. I will say there's some issues with using these clinically yet. I didn't go into this, but for example, if you look at the 27 SNP score that Amgen used, we also tested that in our data set for comparison. And while the curves look somewhat similar, I think in general, the genome-wide score works much better at predicting primary events. Within the clinical trials, they looked pretty similar with their performance characteristics. However, if you look at the two curves and you look at the correlation between those two populations, it's like 0.38 or 0.4. It's not spot on. So I think we're getting slightly different distributions of patients when you use these scores. And I think this was alluded to by the first speaker in Alzheimer's disease, that you can see this depending on which methodology you use, you can get slightly different population distributions. And so when we begin to use these in the clinic for, guiding treatment and things like that, we really need to have that settled. We can't have, depending on which score you use, you're either recommended to take a particular treatment or you're not. I think so going forward, there probably need to be some standards set up by things like the American Heart Association and so forth, so that everybody's being tested with similar tests. I won't touch upon the non-European population one that's been discussed previously. And this has also been discussed too, the idea that these PRSs are lifelong, and particularly in cardiovascular disease, we have a number of, obesity, age is a huge risk factor, smoking status and diet and lifestyle things. Most of those come trigger later in life, but we can certainly measure up a polygenic risk score at a much earlier time point and perhaps take steps to offset some of those risks that come later. So in summary, so patients with high CAD polygenic risk scores receive greater clinical benefit to Alirocumab treatment. The results have now been observed in 2 large PCSK9 outcome studies. The use of polygenic risk scores in drug development may provide a novel genomic tool for patient selection and stratification, and we've certainly begun using them quite extensively as well. Quickly into the acknowledgements, I'd like to thank my colleagues at Regeneron and Sanofi, as well as the academic collaborators on this project. Particularly, I'd like to point out the work Amy Damask did in this study. She was the statistical geneticist that did all the work that I showed today. And also, finally, I'd like to thank the investigators and patients who participated in the study. And I'll stop there and take any questions.
That's great, thank you, Charles. So there's a few questions in the chat, but if I could start, essentially with your PRS, you're just defining a high-risk population. Do you see a different story if you just select on clinical risk scores and selecting the 10% highest clinical risk based upon cholesterol and family history?
Yeah, I think the biggest risk, again, that was shown in the trial was LDL cholesterol at baseline. That was the one that predicted the most. I think there's been some work as well showing that LDL lipoprotein A levels can also have an influence as well. But those are the predominant ones in addition to.
So your gene-based scores would obviously start to drill down much more into, I suppose, what I would think of as a more pharmacogenomic effect, where you're trying to find something that's specific to the mechanism of action. Do you find anything when you drill down?
The problem with that is, if we just ran a drug response trial, and the sample sizes will be relatively small, even in a pretty large sample size. So again, it gets back to this GWAS source data set. And the fact that we're seeing positive results here using a polygenic risk scores because our polygenic risk score is so good for defining cardiovascular events, and that's the endpoint of the study. I think when those two are lined up and you have a big source GWAS data set for the particular endpoint or phenotype you're measuring in the clinical trial, you probably have a much greater chance of success. If it's purely a drug response that, say, you can't find in an EHR data set, type of phenotype would be much more difficult to probably achieve success by running a study with a few hundred people and then doing a GWAS on that and then trying to use that. It's pretty complicated because the sample size is probably. We'll see. We'll try it, but I'm not as optimistic.
Okay, sure. So a couple of related questions, I think, was so Dirk Paul was asking around, do you see a future for PRS guided clinical trials? So you essentially maximize your events through the high risks. And then Dan below was asking, did you calculate sample size you would have needed to observe a larger treatment effect in high genetic risk groups?
Yeah, that's a great question. I actually should have probably touched on that. I see that as a potential, whether it's in this disease or other diseases, where sometimes running the studies, these are events-based trials. So if you can recruit patients who are more likely to have events, you could definitely reduce the size of the study. We haven't done that calculation. I've asked the statistician to do that exactly. So this study was 18,000 people. What if we just did high polygenic risk score patients? How much smaller could this be, and how much shorter could it have been? And so it's a great question. Like, I've asked the same one. I just haven't gotten the data yet on that. I will say that this does bring up issues around your label, your drug label. What would the FDA or the EMA require then? Would your drug only be approved for people with high polygenic risk scores, or would they allow you to just run the study and then still treat everybody? So these are thorny questions that I don't know the answer to that one. So
I guess that might lead into the next question, which is around from Vincent. Is Regeneron working towards using PRS as a companion diagnostic for the drug?
Yeah, for this we're not. It came a little bit late in the life cycle of the drug. It's already out there being sold, and like I said, there's a lot of regulatory hurdles to get over. We would potentially have to run a prospective study. These are hugely expensive trials. It'd be hard. I think that the health authorities probably need to get a pathway for using results like this from very large retrospective studies, and what's the criteria that they require for that, without requiring to go out and run a prospective study that would be several $100 million to run.
Okay, and I think we're slightly over time, but just last couple of quick questions there then so Alex Doney, consensus on PRS for clinical use is probably impossible for regulatory purposes. It'll be forever moving target. How will these ever be implemented in this scenario? So I
Yeah,
sort of,
yeah, and it's a good question. I don't, they're continually getting better and continually changing. I mean, that can be said for a number of things too, though. So I guess, that's to be determined by, I guess, researchers like all of us. Just, I mean, if you think about it, with GWAS studies, when they first came out, it was a little bit of the Wild West. There really was no defining, this is statistical criteria for it. Even in the early candidate gene era, I don't know if many of the listeners remember that, where people would make claims about a lot of candidate genes that all of them fail to replicate.
mm.
And then, it's sort of settled into a statistical criteria required for doing these things. What's the level of evidence one needs before one can claim genome-wide significance and so forth. So we may settle into that with PRSs. There may be sort of, you reach a point where diminishing returns, right? Maybe when the GWAS studies get so big, they really will settle into, adding another 100,000 will not do much to improve the performance of them. And maybe once we reach that point, everybody will settle in on the methods and so forth. And, that's what I'd probably project out. When that point is, I don't know. So we're still in the early phase, I think. So.
Okay, and one last question from Mark. What genetic test do Regeneron use? Is it one per PRS or whole exome stroke genome, so patients could be used in further studies?
Well, right now the sequencing, for rare variants, we do standardly in all our studies. But for this, it's, think about our source data set, the GWAS, you need to have an effect size for each variant associated with the disease. So if it's very rare variation, you don't have that, right? Even if it is playing a role in the disease. So until we get, the effect sizes to put it in the model, you can only go as low as having that in your data set. So, there are certainly known mutations in these pathways, the familial hypercholesterolemia ones. Those are roughly 1 in 250 people. But adding those into the model, other than for those individuals, it probably won't move the needle much for the overall polygenic risk score performance, because there just aren't that many people that you're essentially misclassifying. So.
Okay, great. So thank you very much, Charles. That was fantastic.